Now, post-translational processing of proteins. Translation is not the end of the genome expression pathway. The polypeptide that emerges from this ribosome is inactive and before taking on its functional role in the cell must undergo post-translational processing. In eukaryotic cell, most protein synthesis occurs in cytoplasm. The remainder of the protein synthesis occurs on the rough endoplasmic reticulum. Proteins synthesized on RER, that is rough endoplasmic reticulum, require transport across a membrane before they reach their final destination. It has been discovered that the ultimate cellular location of proteins is often determined by specific, relatively short amino acid sequences within the proteins themselves. These sequences can be responsible for proteins being secreted, imported into nucleus or targeted to other organelles. Let's discuss on protein targeting. Proteins to be synthesized on endoplasmic reticulum have a characteristic leader or signal sequence that directs the synthesis to ER. This process is called as protein targeting. Signal sequence present at amino terminal end of protein. It is usually about 20 amino acid length. Different steps involved in protein targeting are: first one, recognition of signal sequence by signal recognition particle, that is SRP. Second one, SRP ribosome directed protein synthesis into the ER. Third one, completion of synthesis into the ER. Now, let us discuss each step one by one. First one, recognition of signal sequence by signal recognition particle, that is SRP. The proteins synthesized on ER are made in a precursor form. During translation, protein emerges from the ribosome. It is identified and bound by a SRP. SRP is a nucleoprotein consisting of 6 polypeptides and a 7S small cytoplasmic RNA. Second one, SRP ribosome directed protein synthesis into the ER. SRP ribosome complex arrests further elongation of polypeptide chain by blocking ribosome. The SRP ribosome complex interacts with a SRP receptor complex in ER. The interaction of these two complexes, that is SRP ribosome complex and SRP receptor complex, insert signal sequence into the membrane and restarting of protein synthesis. Just prior to restarting of protein synthesis, SRP is cleaved from ribosome using ATP. During insertion of signal sequence into the membrane, the signal sequence binds with another membrane protein called signal sequence receptor, SSR. Third one, completion of synthesis into the ER. While signal sequence is attached on SSR and remainder of the protein is being synthesized, a signal peptidase on the luminal side of ER cleaves the signal sequence from protein. The protein passes through the membrane into the lumen during synthesis. A number of changes in newly synthesized proteins occur in the ER and Golgi complex. The major types of post-translational processing are: first one, protein folding; second one, proteolytic cleavage; third one, chemical modification; and fourth one, indene splicing. Often, these different types of processing occur together, the polypeptide being cut and modified at the same time that is folded. If this is the case, then the cutting, modification or splicing events may be necessary for the polypeptide to take up its correct 3-dimensional conformation, because this is dependent on the relative positioning of the various chemical groups along the molecule. Let us discuss about these concepts. First one, protein folding. When a protein is being synthesized, it may assume a 3-dimensional structure for attaining biological activity. The 3-dimensional folding up of newly formed proteins are regulated by a family of proteins collectively called chaperones. Please see the figure on screen. Chaperones helps to produce the correct spatial arrangement of a nascent protein. They attach a newly formed protein and prevent wrong folding, so that folding is directed only in the correct conformation. Chaperones are diverse family of proteins. Some of them are sensitive to heat, hence called heat shock proteins or HSPs. Production of HSPs increase when the cell exposed to various stress inducing responses such as radiation, free radicals, heavy metals, bacteria, etc. Hence, they are also called as stress proteins. HSPs are classified into various families based on their molecular weight. The main HSP family proteins are HSP10, HSP60, HSP70 and HSP90. In these families, HSP60 is also called chaperone, is well studied chaperone. In this figure, we can see the action of chaperones during translation. Next, proteolytic cleavage. It has two functions in post-translational processing of proteins. First one, it is used to remove short pieces from the N and or C-terminal regions of polypeptides, leaving a single shortened molecule that folds into the active protein. Second, it is used to cut polyproteins into segments, all or some of which are active proteins. A similar type of processing occurs with insulin, the protein made in the islets of Langerhans in the vertebrate pancreas and responsible for controlling blood sugar levels. Insulin is synthesized as pre-pro-insulin, which is 105 amino acids in length. The processing pathway involves the removal of the first 24 amino acids to give pro-insulin, followed by 2 additional cuts which excise a central segment, leaving 2 active parts of the protein, the A and B chains, which link together by formation of 2 disulfide bonds to form mature insulin. Please see the figure on screen, which shows the proteolytic processing of insulin. The first segment to be removed, the 24 amino acids from the N terminus, is a signal peptide, a highly hydrophobic stretch of amino acids that attaches the precursor protein to a membrane prior to transport across that membrane and out of the cell. Signal peptides are commonly found on proteins that bind to or cross membranes in both eukaryotes and prokaryotes.